So I think with that, we will now like to start our panel discussion. We have got a remarkable group of panelists with us, and I think what we'll do is we have a set of questions, obviously, that have been thrown out there by quite a few of our attendees. So what I would encourage our participants to do is please do think about what you've heard, and if you have any comments, then now is the time to certainly ask your questions, and I'll try and get through them as quickly as I can. So I think let's first of all ask all of our speakers present, if you wouldn't mind kindly taking yourselves off mute and also coming back online so we can see you. And obviously, if you aren't speaking, then do feel free to put yourselves back onto mute as well. So let's, I'm going to start with some of the more general questions that we've had. One from Edward Hudgens, who says, hello from Washington, DC. Today, he is revising FDA proposed reform legislation. And if you could reform or completely redo FDA to allow for innovation of discovery and certification process, what exactly would you actually challenge and what would you do? So I'm going to throw that out there for one of you to try and answer and maybe have a think about. And, you know, it would be good to have one of you feedback onto that as well. If you could
think that it will take a
something.
lot of time to actually be able to show the ability to, you know, to prove to the FDA that you can actually dramatically shortcut or some of the processes. So we've seen that obviously with the COVID, but bear in mind that the technologies themselves were, you know, technology that were worked on for sometimes a decade. So there wasn't like a real dramatic shortcut, and this was like a very, very specific emergency. So I think that once there's enough evidence to show that you can actually have, let's say, an excellent prediction for, let's say, toxicity or something like that, and this is shown for a few years, maybe there's a possibility to have a discussion with the FDA, but that's, you know, it's a very tough thing to do. That's my take on it.
Would anybody else like to comment on that?
I was going to tell you that you were muted, but I'll pick up where Dr. Lerner left off and say that there's the primacy of evidence, even when you're talking about AI. And so the approach that we take is in mining the evidence and presenting that evidence. Approaches that have been touched on from other speakers here might be to produce more evidence. And so you'd hate to, especially in clinical circumstances, solely rely on predictions in the absence of that evidence. So it's about finding more of that evidence and refining it and qualifying it. And that's the only way I think you're going to see any change in the FDA's requirements.
Going back to you, Dr. Mark, there was another quick question that we had, which wanted to specifically look at the genome query language. How is AI used with that? Yeah,
sure. We have a different approach to AI where we're not using black box techniques. We have more of a heuristic bottom-up approach. And so undoubtedly, panelists and attendees are familiar with the IBM Watson and their approach to solving this challenge of pulling out genetic and genomic information. We feel like the flaw in that logic was to have enough data to have the patterns just self-assemble and emerge. And we took a different approach, which was to say, understand the myriad different ways that genetic variants can be described and all of the different ways that genes are discussed, and build on that early success and iterate from those early successes, as opposed to taking all of the data and asking agnostically for the patterns to emerge. We found the simplest ways to codify those patterns and built on our early successes, and that's what's culminated in our very comprehensive approach at this point.
Lovely, thank you. Dr. Ha, quick question for yourselves. How long does DHN take to analyze a sequence?
Yeah, so that's a good question. So it really depends on each project. But for example, on average, to identify hit molecules, we can do it as fast as 2 weeks. But it could take a couple more weeks, depends on the availability of the data sets and how well known it is. So yeah, on average, 2 weeks.
And a general question to throw out there as well that was asked earlier is, what about the technological challenges that you might be encountering? It's great to hear about the success stories, but to understand for a lot of people as well who are attending, they may be encountering some of these. It would be good to hear from yourselves about the challenges in terms of the roadblocks with technology and what you have done to overcome them.
I can refer to a few challenges. So first of all, very interesting comment from Andrew. But I have a question to Andrew. So
yeah, sure.
2 weeks. So you're basically selecting a molecule from a known chemical library, right? Or
that's correct. We can. So what we can do initially is to use publicly available data sets, but we have collaboration that a collaborator brings privately available data set that we can also analyze. That also helps to identify novel molecules.
Mm-hmm. Very interesting. So as far as challenges, first of all, we use, as Andrew mentioned in his talk, we do need the 3D modeling or a good structure, and this is still, even though there was the DeepMind breakthrough, people tend to think that this solved all the problems. It didn't. So you can have, in some cases, an excellent model, which is a computational model, and you can have a horrible model. And it's also, it depends on the site on the protein that you're trying to tackle. So there's a lot to do in this respect. That's one challenge as far as we are concerned. Luckily for your company, that's not an issue. And the other issues are around synthesis. So we are fortunate that with peptide synthesis, it's quite, straightforward. So it's a string, basically. You can have some, to play some games with it, like a cyclic peptide with something, a lasso peptide, things like that. But the synthesis is quite straightforward. In many other cases, that's not the case, and you can have anything from a very easy to straightforward synthesis to something that is almost impossible to synthesize. So that's another issue that you sometimes tackle when you try to find an innovative molecule.
Yeah, so I guess I can also add on to maybe one of the challenges that we face. I think one of the biggest challenges is obtaining data that are qualitatively meaningful. So as we all know, technology opens the door to greater data accessibility, but selectively identifying right set of data for a particular project is critical.
Yeah, I agree with that, Andrew. I think data is really a big issue in terms of the quantity and the quality of it. And I think this is where we see, collaborations and partnerships forming. So you can see, pharmaceutical companies forming, for example, partnership with universities who do more research and collect more data, or, partnership with another commercial organization to, try to sort out, the data issue. Because without it, you cannot then, train the model and then, gain that accuracy you need on the outcomes.
Lovely. Actually, Dr. Jamila, touching upon that, I'd be interested to understand what challenges are inspiring you. Obviously, AI promises to improve lives through science and innovation. So with these challenges, what specifically is inspiring you?
AI potential, I'm really passionate about AI, and I think, the potential is significant. Obviously, there are, the good things and the things about AI. I see AI as a tool. So it's the way we use it. If we use it like in a good way, then it becomes good. If we use it like in a bad way or for a bad, objective, then it's bad. So I think, the potential is there, whether in business, whether saving lives. We've seen the theme of AI for good really, expanding over the last few years. We've seen AI, being used to improve, or diagnose and improve mental health. We've seen AI being used to, help with for deforestation, for us. We've seen AI, try to predict earthquakes. We've seen AI in healthcare. There are like so many, outcomes, that is like, good for the overall humanity. But then, you really need to be aware of some of the issues around AI. Obviously, we haven't talked about a lot in here, but like a big issue with AI, apart from the data, is the bias. Again, it's not necessarily AI or the algorithm that is biased, but it's the data that is fed into that could bias. Maybe it's the way, the person who built, or construct that algorithm that could have a biased opinion. So if we've seen, for example, some really example where it went really, really bad, in terms of bias. So for example, Microsoft first Twitter bot, become very racist within 24 hours of interaction with people. We've seen the Amazon recruitment tool that, favored men over women for the same, for technical roles with the same kind of CVs. We've seen it in the US, justice and so on, and also kind of like in some healthcare application. So I think the good, the potential is that it's just like, the way you go about it, you need to be aware of all these. So you include all those things in the design phase, because if you miss the design phase, it becomes very expensive to correct it, when you are, towards the end.
Absolutely. I mean, I was actually going to ask in terms of, are AI applications always successful? I mean, there will be failures, and perhaps other panelists want to contribute towards this as well, is how can we avoid when we are working with AI applications and they just simply don't work?
So maybe, Roxy, if I can build a little bit, and then I leave it to the other speakers to add. So as I already mentioned, AI is not always successful. Obviously, it has been successful, especially in healthcare, in terms of, so it's very good with computer vision, for example. So the use of deep learning has increased the accuracy rate for computer vision accuracy by more than 40% compared to the traditional, methods. So we have some, success example. We have some, failure examples. I mentioned, the bias, in the, Amazon, Microsoft, bot tool. But there is another famous, I think it's called, it doesn't have a name, but like the way, you describe it is the image basically of a husky. So there is a husky with the snow on the background. Then you ask AI, is it a dog or is it a wolf? And then it wrongly classifies a husky as being a wolf. But then what they found out is basically why there was an error, because AI was not really looking at the image of the husky. It was the snow. Because there was the snow, it has automatically, kind of detected that it must be a wolf. And I know, it must, it's just like, kind of like an animal type of, kind of example. But the same thing, can happen with, in healthcare. So some of the application that came up, with reading CT scans for COVID-19, the chest scans. So some of the studies find out that sometimes AI has wrongly identified, for example, a patient, having COVID or not by reading the scan. And some of it, was to do with the position of the CT scan. So for example, if the CT scan was taken with the person, standing up, or if the person was laying down. So for the images where the people were laying down, AI, has given, a high likelihood of, severe COVID. which you think, there is a sense, because if they were lying down, it means they were potentially not well enough even to stand up. But, you can see, it has detected that pattern, and then it has made the conclusion that any CT scan where the person is laying down, it means that it is severe. Where we know in reality, it's not always the case. So you have to be, kind of like aware of those kind of biases, those kind of wrong correlation that could happen in practice. I think
Absolutely.
because of what we do, and then maybe the others speakers can contribute. I think first is to be aware, because if you are not aware of it, you cannot sort it. So the first one is awareness and education around the AI capabilities. The second one is, as I said, the quickest way to sort out things is during the design phase. So during the design phase, for example, you need to have like some ethical principles, some procedure on how to check for bias, maybe some procedure on how you go and look a little bit in terms of the data and try to understand it and so on. But I think the design phase is very, very, very critical.
Naturally, absolutely. Would anybody else like to comment upon that as well?
Yes, I would. Very good examples from Dr. Jamila for computer vision. When I'm looking into drug discovery, so I think two things. First of all, it's a very broad question, but two things that I think are important to take into account. First of all, you're doing a simulation of some kind of reality. So computational power will only increase and the cost would decrease. So even if you reach a failure, you'll probably reach it much sooner and much cheaper than using conventional methods. So even if you take a very grim scenario of a failed project, you'll probably be in a much better situation compared to years of spent research on a wet lab as far as drug discovery. So that's my take on the project, on the question.
Dr. Mark, Dr. Howard, was there anything that you would like to add?
Well, just to summarize, my thinking there is, if you have a failed proposition, is you might reconsider your question, and a different slant there is re-examine the tools that you've applied to seek out an answer. And so if you're continually meeting with failure, you might ask a question with some more precision and increment your way toward the larger answer, as opposed to asking higher level questions that may be challenged by the specificity intrinsic in your data set.
Absolutely, absolutely. Dr. Ha.
Oh, I think everyone covered what I was going to say. So, yeah.
No, I think it's a very good point from all of you, Dr. Mark, to actually review the idiosyncrasies of, particularly when you're looking at AI in a grand scale. As you've said, Dr. Jamila, you have to really think about, first of all, understanding, and then actually looking at the design phase and breaking it down block by block. And it's not as simple as just kind of assessing it as a wide question to throw out there about failures in applications, but really, as you were saying, Dr. Mark, it's highly dependent on each case and each actual design that you're trying to implement, I suppose, as opposed to looking at failures in application. So thank you so much for making that point. We have run over, and I apologize, and I just want to say a really big thank you to all of our speakers. I've been really inspired by some of the conversations, by your work, by the contributions that you are making in drug discovery as well. So thank you so much for that. And of course, if I could ask that in the chat for us to finally add the links to all of our speakers and their profiles and where they work. So I would invite our participants and all of our attendees today to review a little bit further the works and the experiences of each and every one of our wonderful and acclaimed speakers today. Thank you very much to all of you.